transport of ammonia. As we know very well that the ammonia, this one is very toxic for the body. This is very lethal to the body. So whenever the nitrogen containing compounds, the amino acids, they are metabolized in the peripheral tissue. The ammonia is being produced, and as I have told you that this ammonia is very toxic for the body. So this must be transported to such organs which are helpful in the excretion of this ammonia, so that it can not be toxic to us. What I have told you that this ammonia, this is being produced in the peripheral tissue in our body, being produced by the amino acids, by the nitrogen containing compounds. What is that? The ammonia, this is produced by the nitrogen containing compounds by the metabolism of amino acids, and the levels of this ammonia, these levels must be in range from 5 to 35 micromole per liter. If the levels exceed, then this ammonia will be toxic. This ammonia will be toxic, and this ammonia, as this ammonia is being produced in our peripheral tissues, by the nitrogen containing compounds, by the amino acids, this ammonia is being produced in our peripheral tissues. And what is the normal levels of ammonia? That is 5 to 35 micromole per liter, and if the levels exceed from this will be toxic to us. So to avoid this problem, to avoid this situation, this ammonia that must be transported to such organ which are helpful in excretion, and the major organ, and the major, we can say that the part of the body to eliminate this ammonia, that is the liver. So we can say that this ammonia must be transported to the liver. Ammonia must be transported to the liver. Now the question comes that how this ammonia is transported to the liver from the peripheral tissues. For example, this is very important for the skeletal muscles. For example, the ammonia is being produced in the skeletal muscle. Let us say this is the skeletal muscle. This is the skeletal muscle. And here in the skeletal muscle, ammonia is being produced by the catabolism of nitrogen containing compounds as well as amino acids. So this must be transported from the peripheral tissues to this liver, so that this liver can excrete it out from the body to avoid the toxic effects. So there are two mechanisms. There are two mechanisms. To transport this ammonia from peripheral tissues to the liver. And what are those two mechanisms? Number one, that is by the formation of glutamine. Number two, that is by the formation of alanine. So these are two mechanisms, by the formation of alanine as well as by the formation of glutamine. These are the two mechanisms through which the ammonia is transported from peripheral tissues to the liver to excrete it out. Is that clear? First of all, I will talk about the glutamine. This glutamine is very, this is the, we can say, storage form or for the storage of ammonia, and it is non-toxic for the body. So how it is being produced? In our muscles, we have glutamate. With the help of glutamate synthetase, glutamine is produced. Glutamine is produced. Now this glutamine, it will be transported into the blood, and from blood it will be transported to the liver. It will be transported to the liver. In the liver, we have enzyme which is named as glutaminase enzyme. This glutaminase enzyme will cleave this glutamine, which is amide of glutamate, cleave this glutamine and convert it into glutamate plus ammonia. Now the ammonia which was present in the skeletal muscle or the peripheral tissue, from here, from the peripheral tissue, it is transported to the liver, and in the liver this ammonia will be converted into urea, by the help of urea cycle, So this is the first mechanism for the transport of ammonia from peripheral tissues to liver to avoid the toxic effects, or to control the levels of ammonia in the blood. Is that clear? So now we will come toward the second mechanism. If we talk about the second mechanism, again, for example, this is a peripheral cell, peripheral tissue. In the peripheral tissue, the pyruvate that is being produced, the pyruvate which is being produced, pyruvate, this is being produced by two ways. Number one, either with the help of glycolysis, especially anaerobic glycolysis, this pyruvate is being produced, or by the metabolism of succinyl CoA. This pyruvate is being produced, and from where this succinyl CoA is coming, it is generated by the metabolism of leucine and valine amino acids. So what I have told you, that there are two ways for the formation of pyruvate, because we are going to form the alanine from the pyruvate. There are two ways of the formation of pyruvate. Number one, the pyruvate is being produced by the glycolysis, especially the anaerobic glycolysis of the glucose. Number two, the pyruvate is being produced by the metabolism of succinyl CoA. From where this succinyl CoA is coming? It's not coming from the TCA cycle, but it's coming from the metabolism of leucine amino acid as well as the valine amino acids. When the leucine and valine are metabolized, there will be the formation of succinyl CoA, and this succinyl CoA will be metabolized to form the pyruvate. Now, as we know that nitrogen containing compounds and amino acids, their metabolism is producing the ammonia. So for example, we have ammonia over here. When the pyruvate is aminated, or we can say that when there will be the amination of this pyruvate, there will be the formation of alanine. So this alanine is now going to carry the ammonia from peripheral tissue toward the liver, like this. it will come into the blood. When it will come into the blood, it will be transported to the liver, and in the liver there will be deamination. With the help of deaminase enzyme, there will be the deamination of this alanine. When there will be deamination of this alanine, there will be the formation of pyruvate plus ammonia. So here is it, there will be the deamination, and as a result of this deamination of this alanine, there will be the formation of ammonia as well as the pyruvate. Now this pyruvate, what will be the fate of this pyruvate? If we talk about the fate of pyruvate, this pyruvate, it will be converted into glucose. This pyruvate will be converted into the glucose through the gluconeogenesis. Now this glucose will be released into the blood to maintain the level of glucose, or it will be transported to the muscles to give the energy. So these are the two mechanisms for the transport of ammonia from peripheral tissue toward the liver, and this liver is going to control, going to eliminate, going to excrete the ammonia from the body. Why it's going to excrete? To avoid the toxic effects of this ammonia, because we know very well that this ammonia is very lethal for us. Once again, listen to me very carefully that nitrogen containing compounds and amino acids, they are producing the ammonia in peripheral tissues. There are two mechanisms for the transport of ammonia from peripheral tissue to the liver. First mechanism is by the glutamine. And how glutamine is being produced? By the glutamate synthetase enzyme. Glutamate plus ammonia, these are combined together and they form the glutamine. This glutamine is non-toxic. It will be transported into the blood, from blood into the liver. In the liver we have glutaminase enzyme which will convert this glutamine into glutamate plus ammonia, and this ammonia is converted into the urea by the urea cycle, and urea will be excreted out by the kidney in the urine. And second mechanism is by the formation of alanine. This alanine is formed from the pyruvate. And pyruvate, how this pyruvate is being produced? There are two ways. Number one, by the glycolysis. Number two, by the metabolism of succinyl-CoA, which is coming from the leucine and valine. When this pyruvate is formed, it will be aminated and there will be the formation of alanine. This alanine is non-toxic. It will come into the blood, and from the blood it will be transported towards the liver. And in the liver, this alanine is again deaminated. When this alanine is deaminated, or there will be the deamination of this alanine, there will be the formation of ammonia as well as the pyruvate. Again, this ammonia will be excreted out by converting it into the urea. And what about this pyruvate? This pyruvate will be converted into the glucose, and this glucose is going to be used for the maintenance of blood glucose levels or for the maintenance of energy levels. So that's why we can say that this cycle. Now this glucose is again going to the peripheral tissue and again going to produce the pyruvate. So this pathway in which glucose is being converted into the pyruvate, pyruvate into the alanine, and alanine again into the pyruvate and glucose. This pathway is referred as glucose alanine cycle. So these are two best mechanisms to transport the ammonia from peripheral tissue to the liver to avoid the toxic effects.